We did our experiment on temperature and its effect on the turnip peroxidase enzyme system.
Peroxidase was the enzyme used in this experiment, and it was gathered from turnip extract. Peroxidase lowers the activation energy of a reaction and speeds up the reaction time. That is the main purpose of peroxidase. Peroxidase is used in many industries like dairy, textiles, food, and ceramics. In all these industries, time is a very crucial factor. Increasing the efficiency would be a very beneficial way to save money. The question asked was, what temperature is the peroxidase most efficient at? The hypothesis was that the temperature would increase. As the temperature increased, the catalyst would become more efficient. When this reaction was performed and documented in the Dong et al. Article, they found optimum temperature of the peroxidase was most effective at 37 degrees Celsius.
Our experimental design. The turnip peroxidase enzyme system for this experiment calls for the combination of 4.8 milliliters of KPO4 buffer, 50 microliters of glycol, 50 microliters of turnip extract, and 50 microliters of hydrogen peroxide. We had 4 different conditions at 4 different temperatures, 24, 30, 36, and 40 degrees Celsius. For each condition, we did 3 tests for a total of 12 measurements. Each measurement consisted of the same amounts of each reactant, which served as our constant. First, we completed control tests for the reaction. These were conducted at room temperature, or 24 degrees Celsius. All of the reactants were combined in a cuvette and immediately placed in the spectrophotometer. For the next 3 environments, all of the reactants besides hydrogen peroxide were combined in a container and submerged in a water bath set to the desired temperature. Hydrogen peroxide was also submerged in the water bath in a separate container. After 15 minutes, 1 container of solution and 50 microliters of the H2O2 were quickly combined, mixed, and placed in the spectrophotometer for observation. We repeated this 3 times for each temperature, and absorbance was recorded every 20 seconds for 2 minutes, or 120 seconds. The results. This first graph is our line graph, and it is a representation of the change in absorbance of the peroxidase reaction over the 120 seconds measured by the spectrophotometer. There are 4 different lines, each of which represent the absorbance at 4 different temperatures. The blue line represents 24 degrees Celsius, our control test. The orange is at 30 degrees Celsius, the gray is at 36, and the yellow line represents 40 degrees Celsius. To create an environment at these temperatures, the reactant materials were heated to the desired point, combined into a cuvette, and then observed in the spectrophotometer. The control temperature was 24 degrees Celsius, like I mentioned. The concentration and the amount of the reactants, glycol, turnip extract, buffer, and H2O2, remained constant for every trial. The purpose of this experiment was to see how temperature affects enzyme activity, which is represented in this graph by the absorbance rate on the y-axis. We found that after 120 seconds of recording our absorbance, that our enzyme peaked at an absorbance of 0.86 when it was at the temperature of 40 degrees Celsius. This was the highest absorbance rate recorded. Our control group, 24 degrees Celsius, had an absorbance of 0.74. These results are similar to when we measured absorbance for 30 and 36 degrees Celsius. The trend in our data shows that there was minimal or insignificant change at 30 degrees and 36 degrees Celsius of enzyme reactivity. There is, however, a slight increase of enzyme reactivity at 40 degrees Celsius compared to the room temperature control.
Our bar graph showed the absorbance of each trial and each temperature at 60 seconds. The axes are labeled with temperature in degrees Celsius, our independent variable, and the x-axis was labeled as absorbance, the dependent variable. The averages are shown for all 3 trials. In the figure, the bar graph illustrates the average absorbance of peroxidase solutions at 4 different temperatures. The first bar represents our control test at room temperature, 24 degrees Celsius, to which we compared the next 3 tests as we gradually increased the temperatures. At 30 degrees and at 36 degrees, there was no significant increase in average absorbance. In fact, the absorbance is lower, suggesting a lag in enzyme activity. When the materials are heated to 40 degrees, an increase in average absorbance is recorded. The error bars were calculated at a 95 percentile, and they show that there was no significant statistical change and we know that because all of the error bars overlapped. All p-values were over 5%, and that means that the change in temperature did not show any statistical difference.
Okay, so in conclusion, we found that enzyme activity was fastest at 40 degrees Celsius and very minimal at 30 degrees Celsius and 36 degrees Celsius. In our experiment, we found that relative activity was the highest at 40 degrees Celsius, which was similar to Dong et al.'s experiment because they had their relative activity peak at 37 degrees Celsius. One major difference between our results, however, was that our activity decreased at 30 degrees Celsius and 36 degrees Celsius and were lower than our control sample at 24 degrees Celsius, whereas the results in the Dong et al. Experiment were consistent. The independent variable, temperature, would increase catalase enzyme activity because as temperature rises, more hydrogen bonds break, which increase the rate of the reaction. After the activity peaks, the activity of the enzyme decreases. We hypothesized that a temperature increase would help catalyze the reaction to be more efficient. Our results, however, indicate that enzyme activity reduced at temperatures 30 degrees Celsius and 36 degrees Celsius. Therefore, our hypothesis was rejected. Our experiment could be improved by heating the solutions for a longer period of time. The Dong et al. Study heated their solution for 35 hours before measuring the absorbance, while we only heated ours for 15 minutes. If the solution were heated for a longer period of time, then it would be less likely to experience heat loss when transferring the tubet to the spectrophometer. The temperatures would be more reliable and therefore give more accurate results. Our experiment could also be performed at higher temperatures. A greater variety in temperatures would help the experimenters better understand where the enzyme activity peaks. If another experiment were to be done, a greater variety of temperatures and more trials would be very effective.